So, again, it's a pleasure to introduce Emma Davenport. Emma left the sleepy village of Bourne in Cambridgeshire to go to Oxford and do her PhD, then went to Boston and did some time there, and has come back to Sanger Research Centre in Cambridge again. And Emma is going to talk on, let me see, the use of transcriptomic profiling to stratify sepsis patients, which I think will be very, very interesting to many of us. Emma, thank you.
Thank you very much, Eddie, and it's wonderful to be with a live audience and presenting in person. Great, so I'm a group leader at the Wellcome Sanger Institute, and I lead a computational research team. And today I'm really excited to share with you the work that we've been doing on sepsis. So sepsis is a life-threatening condition that occurs when the body's immune system responds to an infection which ends up injuring its own tissues and organs. I can't actually see my slides on the screen. I'm not sure if they're going to come up here or lots of technical issues today. So sepsis is challenging to treat and study for a number of reasons. It can be caused by a chest infection, an abdomen problem, or an infection such as an infected cut or bite. Anyone can develop it, and it kills 52,000 people each year in the UK. Worldwide, unfortunately, a third of those with sepsis unfortunately do not survive. One of the reasons why it's so challenging to treat and study is because there's no reliable diagnostic marker. Current therapy includes antibiotics and organ support, but there are no specific targeted treatments. Despite over 150 clinical trials, we still don't have a good option for sepsis. And one of the reasons we think for these failures is the substantial individual variation that we know occurs in response to sepsis. So the first half of my talk is going to describe some of the work we've been doing over the last few years, looking at inter-individual heterogeneity, looking at the transcriptomic response to sepsis, so looking at gene expression. So back in 2005, the UK Critical Care Genomics Group, including Julian Knight at the University of Oxford and Charles Hines at St Mary's, set up the Genomic Advances in Sepsis, or GAIN study. So this study was for recruiting patients with sepsis as a result of community-acquired pneumonia or fecal peritonitis, which are two of the main causes of sepsis in the UK. And we recruited patients from over 35 intensive care units. And for each of these patients, for which we now have over 1,000 recruited, we collected samples on day 1, day 3, and or day 5 of their ICU admission. We've got lots of different samples collected, so we have collected leukocytes for gene expression, buffy coat to generate genotyping data, and also plasma and urine. So the first study that we conducted was to take 265 of our community-acquired pneumonia patients and generate gene expression data using one of the Illumina microarrays. We used hierarchical clustering and took the two main clusters that we observed as the major drivers of variation in gene expression, and we designated these as sepsis response signature, or SRS1 and SRS2. To understand a bit more about what these two clusters are really highlighting, the differences that we see between the groups, we conducted differential gene expression analysis, and we found around 3,000 of our probes on our microarray were differentially expressed, with the majority of which being downregulated in our SRS1 group. From pathway enrichment, we found that these SRS1 individuals are really identifying those with an immunosuppressed phenotype. But more strikingly, these individuals are also associated with poorer outcome. So when we look at survival, in particular early on, so within 2 weeks of taking that blood sample, we see that those individuals with this immunosuppressed phenotype have a much poorer outcome than those in SRS2. While we were doing this work, we continued to recruit to our cohort, and so we were then able to generate a validation data set, again with the same microarray. And we had just over 100 additional CAP patients, and this time we enriched for the non-survivors, so we had a non-survival rate of around 50% in this validation cohort. We identified a 7-gene set that we can use to distinguish whether a person is in SRS1 or SRS2, and we called SRS group in this new validation cohort, and again we looked at survival, and again we find that those with the SRS1 group assignment have this immunosuppressed phenotype and much poorer outcome. So some of the sort of potential utilities of being able to designate individuals into one or two of one of these two groups is to start thinking about patient stratification and the identification of these individuals with this particular immune response and poor outcome. We can also go down the sort of more basic biology route and sort of try and improve our understanding of the molecular mechanisms of disease, and therefore, we may be able to identify some new drug targets. And then the other side of it is thinking about how transcriptomic signatures could be informative in terms of biomarkers. And this is something that's particularly challenging, as I mentioned, in terms of there's no specific biomarker for identifying sepsis, but also, trying to understand what someone's disease trajectory is going to be, looking at someone's progression, and also then really working out how they're responding to treatment. Can we use gene expression to sort of actually monitor to see if the intervention of interest is actually having a role? So the genomic advances in sepsis study, we are generating huge amounts of data now for this cohort. We've got lots of different multi-omic layers from genotyping, RNA sequencing, proteomics, you name it, we're trying to do it. But my team are particularly interested in linking the genetic information with gene expression. So really trying to look at genetic contribution to the variation that we observe. And so the second half of my talk is going to be focusing on that today. There we go. So the way that we do this is through expression quantitative trait locus, or eQTL mapping. So eQTLs is a way of identifying an association between a genetic variant in the DNA and levels of gene expression. So say we have a SNP, so a single nucleotide polymorphism. In this example, it's an A or a G. And it might be that a transcription factor binds more strongly to one of these SNPs, versions of the SNP than the other. This then leads to an increase in the RNA. And then you get this nice linear association between the number of G alleles that an individual has and then the expression of the gene of interest. So eQTL is something that has been done quite a lot now, and we know from various studies that identifying an eQTL can depend on the context in which you measure the gene expression. So that could be the cell type you're measuring it in, or whether or not cells have been stimulated. And so one thing that we're interested in following up now is trying to identify if we can see eQTLs that are specific to the condition of sepsis. There are challenges in this, particularly finding what the best sort of control cohort is to look at, but that's something that we're thinking about. But we also have an opportunity to actually look at eQTL effects within our cohort. So because we have these two SRS groups, we can look at it within that data set and ask, are there eQTL effects that are modulated depending on which context you're in, whether in SRS1 or SRS2? So this is something called an SRS eQTL interaction. And so we've got the same figure again, but say, for example, the transcription factor that was producing the eQTL effect in the SRS1 group is no longer active in SRS2. We then wouldn't see any difference in gene expression dependent on genotype. And so we would lose that eQTL effect, and therefore we would be able to identify this eQTL interaction. So we have generated a new cohort, so based on the GAINS data again, but for additional patients that have been recruited, we've now generated RNA sequencing for over 600 individuals, and we've got genotyping for these individuals as well. These are all very new, very preliminary results, and I'd just like to thank Katie Burnham, who's a senior staff scientist in my team, for leading on this project. So we have mapped eQTLs in cis, so these are locally acting, so the SNP is in close proximity to the gene of interest. And we find for around 10,000 of our genes across the genome some evidence of an association with a nearby SNP. We've done conditional analysis, so for some genes we find multiple independent SNPs in that gene locus that are associated. So we have just over 16,000 SNP gene pairs that we're then interested in testing for an interaction. Of those SNP gene pairs, we find around 10% are showing evidence of an interaction with SRS group. So this is an example of what one of these interactions looks like. So on the x-axis, we have the genotype of our SNP of interest, and on the y-axis, we have gene expression of the FABP2 gene. Each point here is representing one of the samples from our study, and those coloured in blue have been designated as an SRS1, and those in red as SRS2. What this figure is showing is that those blue box plots have a much steeper gradient, so the eQTL effect is much greater than those with the red box plots. So this is just exactly the same data again, but just a different way of plotting it to really sort of highlight this point. So on the left-hand side, you've got all of the individuals in SRS1, and the right-hand side, all of those in SRS2. And now each point is coloured by their genotype. And you can really see that that eQTL effect is much, much greater in the SRS1 individuals. So we can think about the eQTL interactions as two different flavours, as it were. Those on the left-hand side are dampeners, so they are of a much, much smaller eQTL effect in the SRS1 cohort, and then those on the right-hand side are magnifiers, where the eQTL effect is much greater. And one of our hypotheses is potentially different regulators, so for example, different transcription factors might be responsible for a pattern of, or a selection of these eQTL interactions that we're observing across the genome. And so we are starting to explore that by dividing our eQTL interactions into either dampeners or magnifiers, and then asking, are there any transcription factor binding sites that overlap our SNPs of interest, and are they enriched in one particular type of interaction compared to the other? So it might be that we have a group of dampeners, for example, that are all being overlapping with one particular transcription factor. So for our SRS eQTL interactions, we find about 2/3 of those are magnifiers in the SRS1 group and about 1/3 are dampeners. And we've used the HOMA software to look at the enrichment of transcription factor binding sites overlapping these SNPs. And at the moment, we're finding around 6 transcription factors that show evidence of being enriched in these magnifiers. To try and add some additional evidence and to sort of think about the data that we have, we've also had a look at trying to infer transcription factor activity based on the gene expression data. So because we know the gene expression of the downstream targets of some of these transcription factors, we can try and infer the activity from that, and we've done that using the Dorothea package. And from this, we find around 49 transcription factors. So here we've got all of our SRS1 individuals on the lower half of our heat map and all of the SRS2 on the top half, and it's a little bit difficult to see with these colours, but at the bottom you can see there's generally sort of increased activity of those transcription factors. And around 3 out of 6 from the previous analysis are showing evidence from this particular analysis. So this is just one of the examples, which is ETV4. So you can see if you look at the SRS1 individuals on the left-hand side, there's much greater inferred activity of this transcription factor in that group compared to the right-hand side. And from our eQTL interaction analysis, we found that there were 13 dampener SNPs that overlapped, that were interrupting the transcription factor binding site, and only 1 magnifier. So it's sort of beginning to suggest that some of these transcription factors may be informative. And we need to do some more digging, but a very quick literature search suggests that this particular transcription factor may have links with microbial infection. So what we're really trying to do with this data is to sort of come at sepsis with an angle of looking at the gene expression and really sort of digging into how is genetics contributing to these SRS groups. So we know that these SRS groups are a major predictor of the transcriptomic variation in sepsis. SRS1 identifies a group of patients with this immunosuppressed phenotype and much greater mortality. And it really sort of highlights an opportunity for patient stratification, particularly in clinical trials. There's a lot still that we need to do. We really need to understand, are people predestined to move into one of these groups? Are people cycling through these groups during their disease progression, and that this is maybe a biomarker of those who may be benefiting from particular treatments or particular support? And by trying to do some of the sort of work on the in vivo eQTL interaction side, we're really wanting to try and dig into what the genetic contribution to the heterogeneity could be. Sepsis is quite challenging because there haven't been very many successful genome-wide association studies, so really trying to dig into the genetics is quite difficult, so this is one approach to do that. And finally, starting to think about things like transcription factor binding motifs, thinking about overlapping epigenetic data. All of these sort of approaches, we think, are going to really help us to sort of understand some more of the sort of disease mechanisms of action. So with that, I'd like to thank everybody involved. With all of this research, there's always it's a big team effort, but particularly Katie Burnham, who's been leading on all of this analysis in my group, and my long-term collaborator, Julian Knight, and his team at the University of Oxford. I'd also just like to thank all the research nurses and patients without whom this study wouldn't be possible. Thank you for your attention. I'm very happy to take any questions.